So kind of how this all started off. So I went to Cassia Hall High School in Tulsa, and at Cassia, something that's really special is there's something called the science research team. So that's where high school students interested in science can basically choose a topic that they're interested in and conduct research experiments to learn more about that topic. So we've had students working with cancer, we've had students writing algorithms and doing all kinds of computer science, just doing whatever they love to do. So I joined that team my freshman year, just always being interested in science. And when I began to start thinking about what I wanted to do my project on my freshman year, I was immediately drawn kind of back to the outdoors. And the previous summer, I had gone scuba diving in the Florida Keys, and you could just see all of the bleaching going on, a lot of the, because you see in the pictures like Great Barrier Reef type stuff, all the colors, all the fish. That was not my experience in the Florida Keys, and so that really got me thinking about what's going on in the ocean and ocean acidification and coral bleaching and things like that. So I decided to focus my freshman year on studying ocean acidification. And it was a pretty basic project from the beginning. I wanted to basically see how rising acidity levels in the ocean would affect development of some organisms that are crucial to the whole coral reef ecosystem surviving. So I initially wanted to study how different levels of acidity basically affect sea urchin development. So I designed this whole project. I spent months planning it, on all the different kind of steps on how I was going to get the gametes out of the sea urchins, planning all my different acidities, how I was going to study kind of the development of the sea urchins under the microscope, just getting everything ready. And I got my first real lesson pretty quickly into my scientific research journey, and that is failure happens quite often. No matter how much you plan, science will always find a way to throw a curveball at you and just kind of knock you down a little bit. So 2 weeks before my first science fair presentation, I had no project because these little sea urchins here, when they came in and the time came to do the experiment, I got all male sea urchins out of the entire batch. So you cannot exactly mate sea urchins when you have completely males. So then we were going to try again. So we got a second batch of sea urchins, and they got caught up in a winter storm that we had back in 2016 on their way to Oklahoma, and they were all infertile. So 2 weeks to go before my first science fair, and I had absolutely nothing to present. So after that kind of first little lesson in science, I really kind of dug my heels in, looked at what I wanted to study. I knew kind of early on that this whole water research world had really grabbed me, and I was really interested in it because just water is crucial to absolutely everything, life, the environment, just everything. So I knew I was drawn in and I wanted to do something in that arena. So I had to completely develop a whole new project. And this was my freshman year. So it was a pretty basic level project. So what I ended up doing was just studying different acidity levels and how they affected Lumbriculus variegatus regeneration. So just little water worms, you'd cut them up, put them in different acidities, and study how many segments they were able to regenerate over a few weeks and how the acidity affected that. And then just other basic stuff like mung bean, mung bean root length. So we basically just put little mung bean seeds in acid at different levels of acidities and would measure their roots, how the acidities affected plant growth. And same thing with lettuce seed germination. Very basic stuff, but as kind of my first introduction into the scientific research world, it was really good to do this project to kind of learn how research is done and learn about the scientific method and all the different components that go on there. So this was my very first little science project that I did at the Eastern Oklahoma Regional Science Fair. I ended up getting second. And you'll kind of see how these little science fair presentation boards evolve over the course of the presentation. But there's my first little project. It was great. It got me hooked, and I fell in love with it from this point. And then I fell in love with it so much that in the month between regional science fair and state science fair, I decided to do even more work. As you can see here, I also tested some different enzymes and acidities and just kind of more basic stuff, just learning as much as I could about the scientific research process. So after my freshman year, after that kind of initial project, I knew a few things and I had learned a lot along the way. So I quickly began brainstorming what I wanted to do next. So I knew that I really liked water research and environmental research, but I mean every science classroom studies acidity and what they do in high school. I wanted to do something that nobody had ever done before. And I also didn't want to just talk about a problem like acid is bad. If we have it in our water, it's going to kill plants. Yeah, that's great. But I also wanted to see what I could do about problems, see what I could try and do to come up with, see what I could come up with to fix those problems. And so, sophomore year, I really just kind of started going over the top. Every free minute I had, I was reading as many articles as I could, reading scientific studies, just seeing what was out there that had not been studied that I could get into and see what I could figure out. So what I came upon was fire retardants, like all the red stuff you see on the news coming out of the plains with all the wildfires going on. I figured out pretty quickly that not a ton of research had been done as to what those fire retardants do to our water ecosystems in the environment. And another component that was very interested to me, as I mentioned in the beginning, I love the outdoors. So I wanted to be able to actually go in the environment, get samples from contaminated water systems, and really just study what's actually happening in the environment. Because as great and as nice as simulations can be, nothing's really going to replace the real thing because the environment just has so many factors. There's no way to really replicate that in a laboratory setting. So I also knew I wanted to do field research. So then a project kind of began to come together. And so my sophomore year project really kicked off with a trip I got to take up to Colorado to actually study a stream in an actual live wildfire. The EPA up there actually helped kind of set that up, and we had a few people from the EPA kind of walk us around and help us get the lay of the land. And so you can see me in this picture in the wildfire. We were able to take water samples from streams where fire retardants had been dumped only a few days before. So that was really great to be able to actually get out there and see what it's like and get the real water that had been exposed to fire retardants. And I'm sure many of you probably recognize something called the Water Quality Index. So this is everything I was looking at. I wanted to see every little component of how the fire retardants were affecting the streams in the environment. So I was looking at dissolved oxygen temperature, pH, turbidity, conductivity, velocity, fecal coliforms, nitrates, phosphates, just every possible little component of a water ecosystem. And yeah, here's just a picture of me in the lab working some water samples. And so basically what I figured out from the initial kind of testing I did in the field is that the fire retardants are ammonium polyphosphate based. So they're extremely dangerous to causing excessive eutrophication in the stream, which, as you know, too many nitrates, phosphates, everything's just going to grow, algae blooms, that kind of stuff. And then once everything is just grown as much as it can and all those nutrients are gone, everything just dies and nothing is left in that stream. So it really absolutely kind of destroys the entire aquatic ecosystem. And so that's what I kind of figured out. That's kind of like my first year project where I was just like, okay, here's the problem. But then I wanted to take that next step and start looking at, okay, how do I fix this problem? So I wanted to see how the fire retardants directly affected the different species in an ecosystem. And so to do this, I kind of had to take a little dive into engineering and design what I called the simulated stream flow system. And so that's what this looks like. So there are 3 in this picture. Each color of water is its own system. And this was really important because, I mean, in the ecosystem, usually kind of in the streams and ponds and everything, you're not going to find a lot of just stagnant water. Water is moving. So if you have a fire retardant that gets dumped upstream, that stuff's going to travel all the way downstream miles and miles and miles. And so any attempt to try and fix the problem also has to be able to work while water is moving. which is a little bit challenging. So what I was able to do here is just design a little system to be able to test some of my potential solutions to the fire retardant problem that I found by creating a moving water system. So you can kind of see there are little 3 aquarium tanks connected together with little PVC pipes and little pond pumps and buckets at the bottom, and water would basically be pumped from the bucket at the bottom to the top and then flow down the little streams, just creating some moving water to kind of help resolve that issue. And so in this picture is actually some of my trials. So what I wanted to do, as I mentioned, is see what these fire retardants would do to organisms. So on the far left of your screen, you'll see my control tank. And in there you have some rocks, some allodia, and some little just pond snails. I use pond snails as an indicator species because there's organisms like little macroorganisms that are so much smaller than snails, but then there's also like the fish and other organisms that are bigger. So it'd give me kind of a medium gauge to see how fire retardants are going to affect different species across the food chain and across different sizes of the stream ecosystem. And so the controls on the left, and then in the middle, you can see how cloudy that is. That is actually less than 1% concentration of the fire retardants that are dumped out of the planes and everything. And within less than 24 hours, all the snails in that tank completely died. And then on the right is actually less than 0.5% of the on the far right is actually less than 0.5% of the concentration of fire retardants that's dumped. And within 2 days, all of those snails died. So from a toxicity standpoint, you can kind of see what was going on. And that kind of just gave me more information as to where I wanted to go with a remediation attempt for these, basically just removing all those ammoniums and phosphates from the water that would cause the eutrophication. So there's just kind of another look at that stream system. And so, yeah, as I mentioned, concentrations of less than 1% of the fire retardants killed all of my snails and all of my tanks. But the control system, I mean, they were all living. So there was clearly a problem with a very low, very, very low concentration of the fire retardants. So next, I really wanted to focus on the best way to remove these fire retardants and these compounds from the water systems. And so as opposed to going for like a more mechanical approach or a chemical approach that could have kind of longer lasting or unknown effects on an ecosystem, I turned to bioremediation, basically using other living species to do the remediation so that there's really no, or at least as limited of an impact on the environment as possible as we're trying to clean this stuff up. We're trying to clean it up and not really make it worse or introduce anything worse into the ecosystem in the process of trying to clean one thing up. So I needed to find my organism, what's going to be able to break down the ammonium and use the phosphate to basically break down these fire retardants and turn them into something that's still going to remain in the water for the most part, but that's not going to be toxic. So I turned to cyanobacteria. Cyanobacteria is blue-green algae, and what I needed the cyanobacteria to do is nitrification, breaking down the ammonium in the fire retardants into nitrites and then into nitrates, which of course are nutrients and pretty good for the stream in moderate amount, of course. And so actually what I was able to do is, as I mentioned, the fire retardants are ammonium polyphosphate based. So what the cyanobacteria do is they use a lot of those phosphates already in the fire retardant. They use those up and they're able to use them to perform the process of nitrification at an increased rate. And of course, the end goal here is to prevent eutrophication, which is the main problem. And so the next step in kind of designing my remediation attempt was, now that I had my organism, I didn't want to just introduce a random organism into the environment. Of course, that can cause just so many chain reactions, so many problems. It could be an invasive species. It's a non-native species to the environment potentially. So I needed to figure out a way to basically contain all of my algae and not directly introduce it into the ecosystem, but still allow water and all of those ions to pass through and get access to the algae. So what we came up with here is what's called a sodium alginate bead. Sodium alginate is like this clear jelly-like polymer substance. And when you kind of mix it up and add algae throughout that jelly-like substance, and then suck it up in a little plastic pipette and drop like kind of make little droplets into calcium chloride. The calcium chloride seals the algae and that jelly-like substance into a little almost like jelly bead. If you've heard of like boba, like the boba tea, it's kind of like the little boba balls almost, so that the algae is completely sealed within that little alginate bead, and no cells are going to be able to escape the little bead. But at the same time, water containing all of those ions that need to be cleaned out can pass through the bead. And so as water passes through the little sodium alginate bead, the algae inside are able to do that nitrification process and break down that ammonium into nitrates. And so here's a little picture of what those beads look like. So on the left, here's what the beads look like going in. They aren't very green, although there is algae in these beads. But then after about 24 hours, this is what they looked like. So you can see that the algae were able to use the phosphate from the retardant to really begin to grow and start that nitrification process, and just kind of make the beads perform better. So as the beads are doing their thing, they're able to also get better and increase their rate and increase their success as time goes. So it was a really cool system to be able to develop. So then it was time to test if they actually work. So I took that same simulated stream system that I originally had the snails in and the different concentrations of the fire retardant, and I put them in. And after 1 week of allowing the beads to be in those simulated stream systems with the water kind of circulating over that week, I put more snails back in to see, okay, did they work? And after 1 week, all of the snails lived in every stream that I tested and all of the concentrations that I tested, showing that the nitrification of my little symbiotic, my little beads worked. And it was as I mentioned, the same concentrations that killed the snails the first time around. So it was a successful system that I was able to start developing. And also I was able to do kind of a few different tests with fire retardants to just kind of better understand the general picture of what's going on. So I tested different concentrations of fire retardants with brine shrimp eggs and their hatch rate, amphipods and copepods, little microorganisms in the stream, of course, kind of studying different levels of the food chain, because if one level, especially the most basic level, kind of down with those microorganisms, gets disrupted and killed off, I mean, your whole ecosystem's in complete free fall. And then also some different bacterial tests. I was beginning to learn a bit more about microbiology and get involved and see what was going on with the bacterial components of the aquatic ecosystems as well. And so that's what this project looked like. This was my sophomore year. So you can already kind of tell a difference between that first little science project board that I showed you and what I was able to do my second year. I was just so involved with it. I loved every single second of it. And so I began to really be able to get some great opportunities to present my research and what I was doing. So I won first place at a regional science fair. I was invited to apply to the Oklahoma competition of the Stockholm Junior Water Prize, and I actually didn't even win this year. Didn't even make it to nationals, but that's okay because that got to come later. And yeah, it was just such an incredible experience. This project is really what took my interest to the next level. That first year got me hooked, but this sophomore year project just showed me all the different avenues that are open in water research and all the different opportunities. And so that year I also got to go present my research at the National Junior Science and Humanities Symposium in San Diego. And I actually was able to win that, so that was crazy. And also at the International Sustainable World Engineering, Energy, and Environmental Project Olympiad in Houston, where I got second at that competition. And then I also got invited to go and be an observer, so not compete, but kind of just go see the International Science and Engineering Fair, which is the world's largest pre-college science fair. And so that basically has the top 2,000 young scientists in the entire world with their own original research, and I got to go and see it. And so that was a really incredible opportunity. And those are two of my teammates from Cassia Hall who were both actually competing that year. So that was incredible. And then I also got to go present at the American Junior Academy of Sciences. And yeah, just so many incredible opportunities. Just this whole year was about figuring out what I love to do and starting to meet as many incredible people as I could. And so from those first 2 years, I was ready to do something big. I was ready to take it to the next level. And I knew what I loved at this point. I knew I loved water research. I knew I loved being in the field, and I knew I loved finding solutions. So it was time to really just tackle a whole new problem head on and see what I could come up with. And so now we're going to get into kind of a bulk of the research and the project that eventually took me to Sweden. So this is my junior year project, and that's quite the title, I know, but we'll go through it as best we can. So my whole junior year project focuses on heavy metal bioremediation, specifically with acid mine drainage, which some of you probably know. Acid mine drainage and the abandoned mine sites are all over this country. And it's a really big problem because not only is all this runoff coming out of the mines containing heavy metals on its own, but it's also extremely acidic. So as all that acidic water goes over all the chat and all just the stuff that was pulled out of that mine, it's dissolving more and more and more heavy metals into the water system. And as I mentioned earlier, mines are pretty high. So when you have all those heavy metals just coming close to the water sources, those heavy metals are going to be taken miles and miles and miles away, just contaminating the entire water ecosystem. So, I mean, of course, thousands of studies have been done over the years just showing heavy metals are bad. Of course, like lead, nickel, they're all just terrible. They've been linked to cancer. They just kill everything. And so the Environmental Protection Agency, EPA, established the Superfund Cleanup Program to begin to try and tackle a lot of this heavy metal contamination in water ecosystems, and especially around these abandoned mine areas. And so with a little research I did, I found out that, as you can see on the left, there are over 500,000 abandoned mine sites in the entire United States. But with current funding, there only 1,332 of those are actually EPA Superfund sites. So the vast, vast majority of the abandoned mine sites leaking all this heavy metals into our water supply aren't even really being cleaned up. They're just out in the wilderness somewhere, out in the middle of nowhere, just leaking all this stuff. And a big reason for that is because of funding. Of course, there is limited funding, and these research efforts and cleanup efforts are just so expensive. And so also on the other side of that, a lot of kind of the current cleanup methods aren't necessarily low impact. There's a lot of more permanent facilities in places, and that's because just the damage is so bad. It's so expensive. The only way to really clean this stuff up is to really kind of have a permanent facility and really just treat the water as a ton. And so really I was able to identify the problem as being there's not a cost-effective, low-impact method of heavy metal remediation that can easily be implemented into contaminated water systems. So this is not necessarily even, this is going beyond the Superfund program. How are we going to be able to clean up these 500,000 abandoned mine sites that are putting heavy metals everywhere? It's starting to get into our drinking water, our aquifers, the environment. It's a major problem that's not talked about enough. So, as I mentioned, I like finding out more about the problems, but then also figuring out how to fix the problems. So I designed a 5-phase approach to kind of begin to tackle this. So first, I started in the field. I looked at 2 mine sites, one in Oklahoma, Tar Creek, it's a Superfund site, and one in the mountains of Colorado, it's called Pennsylvania Mine. And so I selected different sites kind of along water systems where abandoned mines were leaking all these heavy metals into the ecosystem, and I collected water samples specifically to collect bacteria. The key to my system this time around was using bacteria, because a lot of the the systems, you'll see some pictures in a couple of slides. I mean, the water is so toxic, nothing's able to even live there. The only thing that can possibly live is just bacteria. There's no plant life, no animal life. It's just barely the bacteria that are so resistant that they can survive in those environments. So I did kind of the field research, did the water quality indexes at all those different mine sites, and collected a ton of water samples. So then in phase 2, I would isolate a bunch of different bacteria from those water samples directly from the environment where the bacteria are already heavy metal resistant and potentially possess some remediative properties. So up next, after phase 2 there, of course, I had at one point over 250 individual bacteria isolates. So I needed to, I mean, making a system and trying to pick and choose randomly a bacteria out of the 250 that's actually going to work. That's just crazy. So in phase 3, I came up with some screening protocols to figure out which bacteria had the best potential to actually be able to remediate heavy metals. And then in phase 4, it was important that I knew what the bacteria were so that I could learn more about their properties, learn more about how to pair them and make the most successful system as I could. So in phase 4, I performed what's called a 16S ribosomal subunit analysis, basically sequencing the DNA of the 16S gene in these bacteria, and then matching them with sequenced 16S genes to be able to kind of match what I had to already identified bacteria to get a better idea of what they were. And then finally in phase 5, once I'd figured out what I'm actually working with, I would design my remediation system, which we'll get into here in a second. And as I mentioned, I mean, there's so many heavy metals, I couldn't really test them all. So in this part of the project, I focused on 7. I focused on lead, zinc, iron, cobalt, manganese, chromium, and cadmium. So phase 1, as I mentioned, field research. As you can see kind of over here, that right there is Pennsylvania mine. As you can see, nothing is living in that water. And it was actually, a lot of it was frozen. It was September and it was pretty high up. And then down here, you can see Tar Creek, again, just a completely toxic waste zone. And so I studied many different places along these streams. It was important to me that I studied upstream from the point of impact where the mines were leaking these heavy metals and this acid mine drainage into the water to see especially how drastically the heavy metals and acid mine drainage really disrupted the water quality of the streams. I, of course, tested the mine sites themselves, water around them, to kind of see the highest amount of heavy metal concentrations in the most toxic areas. And then I also tested sites pretty far downstream, miles and miles downstream, to kind of study how far the metals were able to travel and how concentrated they stayed at different increments away from the mine site, and to kind of study the devastation way downstream from the point of impact. And so after I kind of did that whole process, I went back to Oklahoma, back to my high school lab, and I started extracting my bacteria. So that's basically just taking my water samples, putting them on a nutrient auger plate, putting them in an incubator, just seeing what grows, and then picking the little bacteria colonies and putting them in little nutrient broths, little vials of nutrient broth to let them start growing. And so by the end of that process, I had 250 individual bacterial isolates, and that is a lot. So I needed to narrow down the 250 to the select group that would be best suited to actually be able to remediate heavy metals. So, of course, to be able to remediate heavy metals, you need to be able to survive in heavy metals. So my first process I developed was it's really simple. Just taking each one of those 250 bacteria and adding them to a nutrient auger plate that contained 20 parts per million of lead, and then a different plate that contained 100 parts per million of iron, just to see if they were even resistant, if they could even survive. So 189 of my 250 were at least resistant to those levels. And so with those 189, the next step was really the key to my whole system working. So a lot of bacteria when they grow, they can form what's called a biofilm. So that's when all the different bacteria cells clump together and kind of act as a single unit. And when a lot of these bacteria cells form these biofilms, those biofilms can then work and remediate heavy metals. They can intake the heavy metal ions into the biofilm or stick them on the outside. And so a lot of the reason you don't see this happening is because you need a lot of nutrients for that to happen. And then these just completely wasted streams with all this acid mine drainage, you have no nutrients for that to happen. So the bacteria can survive, but they're not going to be able to attain that level of growth where they're able to form a biofilm. So next I needed to see which of these bacteria can actually form a biofilm in extremely high concentrations of heavy metals. So I went with 100 parts per million of lead, iron, and cadmium, and I added each of my 189 bacteria strains to each of those metals, 100 parts per million, let them grow, and then I stained them with crystal violet and used a microplate reader to measure the absorbance of, so passing light through the crystal violet dye that stained the bacterial cells. So of course, more bacteria cells would have resulted in more of the dye sticking to those cells. So a higher absorbance basically means a higher amount of growth occurred and a higher potential for biofilms growing. And so I selected the 24 bacteria out of that group of 189 that showed the greatest potential based on those criteria. And from that process, with those 24 bacteria, I needed to figure out what they were. And so I won't go into a lot of the details on this like super detailed process, but basically a 16S ribosomal subunit analysis takes a single colony of my bacteria, cuts out the 16S gene, replicates it hundreds and hundreds of thousands of times with PCR, and then you kind of just check, make sure the gene's been correctly amplified with electrophoresis. And then I got to use a super fancy piece of equipment at the University of Tulsa called the gene sequencing machine. And that literally, as you can see right here, it literally gives you the A, G, C's, and T's of the DNA. Like you can have the exact DNA sequence of that gene. So with an online database, I was able to match my bacteria to what had already been identified. And here's a little phylogenetic tree of what I found. In my final group of 24 bacteria, I had 6 different genera: Xanthanobacterium, Colomonas, Flavobacterium, Pseudomonas, Arthropacter, and Bacillus. So I tried to go for as wide of a range of bacteria and genera and species as possible, because as I was trying to figure out the system, I mean, one bacteria might work really well on one heavy metal, and it won't work on another one, but then you have another bacteria that is great at remediating that heavy metal. So that was important to also have a pretty diverse group. And so then it all came down to designing my actual remediation system. And it all kind of rested on this theory that I developed that uses both algae and bacteria in the remediation effort. So, and it's also inside one of those sodium alginate beads. So that's one thing that I picked up my sophomore year that I kind of continued to develop in this junior year project. So the whole theory behind it is your algae in your little bead is going to grow and photosynthesize, producing nutrients and dissolved oxygen that then allows the bacteria to grow and begin to form those biofilms. And as those biofilms form, they begin to use those mechanisms that they have to remediate heavy metals. And as the heavy metal concentration begins to go down in the water, thanks to the biofilms, the algae begins to grow better because it's a less toxic environment. And as the algae keeps growing, the cycle continues to repeat. So it's almost like a little self-sustaining remediation system. And so here's kind of a breakdown of what that looks like. So over here you have your sodium alginate bead with both your bacteria and your algae on the inside. And those bacteria will begin to form those biofilms, and through the processes of bioabsorption, bioaccumulation, and bioassimilation, as heavy metals, kind of very similar to the process I described, all these cells are completely contained within this little jelly bead, but the water containing all these heavy metal ions can pass through, and they'll begin to clump around and remain within the bead. And so the time came to test it. So the moment of truth. So I made all of my sim beads. I actually made a bunch of different systems. I wanted to test each genus of my six genera that I discovered. I wanted to test each one separately to really kind of figure out which genus is good with which metal and that kind of thing. And so now we're going to get into the results. So right here you have lead. And so on the left of all these graphs we have coming up is the control both before and after my trial. So I started with 5 parts per million, and then all these other bars, the dark blue bar represents the initial concentration, and the green bar represents the change after my trial, after uh, my bacteria algae beads have been exposed to these heavy metal concentrations. And so you can see there was quite an extreme drop for lead, but then the results just kept getting better and better and better. This is iron. This is cadmium. This is zinc. This is manganese. This is chromium. And this is cobalt. And so all that information is kind of summed up in this table right here. All of the dark green boxes indicate an over 80% decrease in the heavy metal concentrations in just my short little trial period. And so that's extreme potential based on what's being done with multi-million-dollar systems and just what I was able to accomplish just with using these species that I was able to extract from the environment itself, give it the little environment that it needed to start remediating, and go from there. And so kind of after this junior year, I mean, the results were extremely encouraging and motivating to continue my research later on in my senior year. But kind of my general conclusions from this initial project was, I mean, EPA's $3.3 billion trying to clean up less than 1% of all the abandoned mine sites in the entire United States. Like you can see right there, 99.997 of the mine sites in the United States are not regulated. And my whole system cost $10 to make. And with the majority of the systems removing over 80% of the heavy metal concentration, the results were just so promising because it's low impact, it's very cost effective, and it's so successful at doing what it was designed to do. We could go out to these mine sites that aren't part of the Superfund program and begin to clean them up. And so here's just a few pictures from all of my hours spent in the lab. Lots and lots and lots of work, but I loved every minute of it. Here you can kind of see a before and after. There's my junior year board on the right, a little bit of the planning that goes into it. And then I was able to win our regional science fair, and that actually sent me to actually be able to compete at the International Science and Engineering Fair, which was an incredible opportunity. It was in Pittsburgh that year. So there's my team. As I mentioned, I'm not the only one who was able to do this through high school. Everybody kind of had their own thing and did their own little science projects. And then I actually won Best of Fair at the Oklahoma State Science Fair, and got to again go present all across the nation, meet the most incredible people. And then the big moment, one of the big moments, I won third place in environmental engineering at the International Science and Engineering Fair. That same event where I was just in awe, just going and looking around at all the different people just the year before. And so to win third in the entire world, that was incredible. And then came the Stockholm Junior Water Prize. So I was invited to submit to the Oklahoma competition, and I was selected as the Oklahoma State winner. And I got to go to the national competition, and the judging was based on a 20-page research paper, a 3-minute video containing basically 6 months of work down into 3 minutes, and then interviewing with judges who are the top water professionals across the entire country. So there's some of my judging. And then I won it, and I still remember it blew my mind. Like at first I did not think they said my name. I remained sitting when they said my name. And somebody next to me had to be like, that's you have to go up there. It just blew my mind. And then Actually, I also got to be featured on a TV show there. That right there is the grandson of Cousteau, the inventor of scuba diving. So it was also kind of cool to see it come full circle. My initial project with the sea urchins freshman year that completely failed, that got me, I got into that because of scuba diving. And here I am talking to the grandson of the guy who invented scuba diving. So that was kind of a cool full circle moment. And then I got to go to Sweden, represent the United States. It was such a cool experience. We got to do some good sightseeing around the old parts of Stockholm, of course, IKEA, because you're in Sweden, and just met the most incredible people. It was awesome. And one of those people being the princess of Sweden, Crown Princess Victoria. And then on the way home, we stopped at Iceland. Lots of great pictures there. And then came my senior year.
And we've got about 1 minute to wrap up. I hate to interrupt, but
Okay, sweet.
okay.
Sweet. So what I spent my senior year working on was basically just figuring out how to take this system to the next level and how to implement it into the environment. So I took my heavy metal system, I added 3 more heavy metals, I added arsenic, I added agricultural runoff, I added fracking, I added oil spills. And then I also 3D printed and designed where is it? There it is. A little buoy system to actually deploy these into the ecosystem. So that's what I spent my senior year on. And I was able to win second place at the International Science Fair that year and just keep going at it. So yeah,